Welcome to AP Biology. I want to talk today about endocytosis and exocytosis. I have a quick video here that just want to turn the volume off for it. So what's happening here is some material is coming over to the cell membrane, and the cell membrane sort of makes this little pocket and pulls it inside. And so you can see a little vesicle actually starting to form. So this is outside the cell. Now we're going into the cell. And here's that vesicle that's formed that brought the material into the cell. And the opposite can happen too. So here's a vesicle that's coming from the Golgi, and it fuses with the plasma membrane, the cell membrane, and it's releasing its contents outside the cell. So in this video, we're just going to define those things and then some examples. So exocytosis, think exit. This is materials that are leaving the cell. And so a vesicle from the Golgi body comes over, it fuses with the cell membrane, and then this vesicle now is part of the cell membrane itself, which is kind of cool. And so anytime you need to move a lot of a material, so maybe one neuron is sending impulses to another neuron, and so the impulse is going along the neuron, the nerve fiber here, and then it releases its materials out. And so it must release a whole bunch of those neurotransmitters at once that will then go and talk to these other neurons that are nearby. So anytime you're moving a lot of something, or anytime you are moving like a big molecule, like maybe you need to bring collagen out of the cell, maybe you're like a cartilage cell or something and you need to have a kind of big proteins outside, you might use exocytosis to release them. Here's an exocytosis video. I just like how it shows how the cell membrane of the vesicle fuses with the cell membrane of the with the plasma membrane, the outer membrane. Okay, endocytosis is the opposite. It's moving stuff in, so you can see the cell membrane here, coming in and making a little pocket, and then more so, and then more so, and then the proteins here kind of will close this up and pinch it off, and you'll have a vesicle. There are three types of endocytosis. This term, sorry, it's coming up. So this is just an animation showing you taking materials into the cell, endocytosis, endo for inside. Okay, so the first one is phagocytosis, and that means bringing in bigger, like a solid material. So for example, bacteria. So here's a white blood cell taking in a bacterium. It's a type of endocytosis called phagocytosis. And this would be a phagocyte or a vesicle right here. And what typically happens if you have live bacteria inside your white blood cell is that they'll fuse with a lysosome, and then the digestive enzymes here will break down the bacteria. All right, another one is pinocytosis. We don't really use this term all that much, but in pinocytosis, molecules are taken up when just the fluid outside is sort of brought in through this endocytosis. And then finally, the last one does come up. I want you to know this one. Receptor mediated endocytosis. This one is specific. So vocab word first, ligand. A ligand is any molecule that binds specifically to a receptor site of another molecule. So here's the receptor, and there's one here too. All these red triangle-y things are the receptors, and the purple thing is the ligand. So this green thing would be a ligand for a different receptor. So this attachment here between the ligand and the receptor is very, very specific. So if this is a receptor for serotonin, then this would be a ligand. This ligand would be serotonin. So it's a good match. So what happens is on your cell membrane, you might have all of these receptors spread sort of out. And then once they have their ligand attached, they're all going to kind of wiggle together, and you'll see them later kind of bunched up sort of close to each other. And so that's in order to get this particular ligand into the cell. So why might that happen? Let's say you've got a neuron. And so the neuron gets stimulated, let's say, and it sends an impulse this way. And when it does that, the impulse gets to the end here. And let's say this one is releasing serotonin. So that serotonin is going to diffuse across the distance there. It's actually a much smaller gap than I'm showing you here, but here's the second neuron. And this ligand, this serotonin, will diffuse across the little gap. It's actually much smaller, and attach to this receptors on the second neuron, maybe to cause that neuron to fire. But you should only have this serotonin out here for a little bit of time, because you want to stimulate the next neuron, but then stop stimulating it, so that if you keep sending more and more serotonin, great, continuously tell that neuron what to do, but you don't want it being stimulated when you're not telling it to get stimulated. So you've got to get rid of it somehow. So there are a few different ways to get rid of it. One of them is for these other cells, these glial cells, to pick them up. So glial cells. So, or neuroglia cells if you want. So here's a glial cell, and it's going to have receptors for serotonin. Oh my goodness, for serotonin as well. And so these receptors will also take the ligand, the serotonin, up, but then they might take it in and just destroy it so you can get rid of it. And that's my story. Bye.